Hello, everybody. In this lecture, we're going to talk about RNA. RNA is closely related to DNA, but it has several important differences. We will examine the structure of ribonucleotides, compare RNA and DNA, and discuss the major types of RNA involved in protein synthesis, and explain why some organisms can use RNA as hereditary material. All right, so by the end of this module, you should be able to describe the biochemical structure of ribonucleotides, describe the similarities and differences between RNA and DNA, and describe the function of the three main types of RNA used in protein synthesis, as well as explain how RNA can serve as hereditary information. This here we see that we see a ribonucleotide. There are three major components, very similar to what we saw with DNA. We have the phosphate group, the ribose sugar, and the nitrogenous base. RNA nucleotides are very similar to DNA nucleotides, but the only real difference that you'll see is that the sugar is different. Ribose contains an OH group on that 2' carbon, while DNA lacks this oxygen. That extra oxygen makes RNA more chemically reactive and generally less stable than DNA. RNA contains 4 nitrogenous bases: adenine, uracil, cytosine, and guanine. Now, notice that RNA uses uracil instead of thymine. During RNA synthesis, adenine pairs with uracil. The image here shows the structural differences among these bases. You can see at the top on the right hand side you have uracil, and on the top on the left hand side you have thymine. Adenine and guanine are, again, those larger double ring purines, while cytosine, uracil, and thymine are smaller single ring pyrimidines. Now, RNA and DNA both are molecules that are nucleic acids made from nucleotides. Both contain phosphate groups, sugars, and nitrogenous bases. Both also carry genetic information using sequences of those bases. Even though RNA and DNA differ structurally, they still use a very similar chemical language, the language of nucleotides or nucleic acids. Now, there are some differences though. RNA contains ribose sugar, while DNA contains deoxyribose, and RNA uses uracil, while DNA uses thymine. DNA is usually double-stranded and forms a stable double helix, while RNA is usually single-stranded and more flexible. Because RNA is less stable, cells often use it as a temporary working copy of genetic information. So the way to think of it is that DNA is very stable. It's going to stick around for a long time. You don't want it to go anywhere. RNA is more reactive and less stable, and so it functions really well if you don't want it to be sticking around all the time. This will make more sense when we start talking about making proteins a little bit later on. Now, here we have messenger RNA. mRNA carries genetic instructions from DNA to the ribosome. The sequence of bases in mRNA is organized into codons. Those are little triplets, which specify amino acids during protein synthesis. We'll talk more about this later. You can think of messenger RNA or mRNA as a temporary copy of a gene that the cell uses to build proteins. Also, just as a heads up, I will interchangeably call this mRNA, messenger RNA, the message, or sometimes the transcript, right? So all those words mean the same thing here. Now we have ribosomal RNA or rRNA. Ribosomal RNA combines with proteins to form ribosomes, which are the structures where proteins are assembled. rRNA is not just structural, though. It also helps catalyze peptide bond formation between amino acids during protein synthesis. So they act as what are called ribozymes, which are RNAs that basically have catalytic activities that are in the ribosome. Here we have transfer RNA or tRNA. tRNA acts as an adapter molecule during translation, the process of making proteins. One end of the tRNA carries a specific amino acid, while the other end has an anticodon region, which is a triplet base that pairs with a matching codon on the mRNA. So when we talk about protein synthesis more, we'll talk about how that works. But essentially, you have an anticodon on your tRNA and a codon on your mRNA, and they bind together briefly using those hydrogen bonds. And this helps ensure the correct amino acid is added to the growing protein. Now, this is just a little hint because we'll get into this a bit more. It explains that, we see that RNA can function as hereditary material. Um, Now, most organisms use DNA as their genetic material, but some viruses and some other acellular entities use RNA genomes instead. RNA can store information in its nucleotide sequence just like DNA. Um, and the other cool thing is that RNA can also perform catalytic functions, which is why some scientists, why scientists developed the idea of the RNA world. This hypothesis proposes that early life may have relied primarily on RNA before DNA and proteins evolved, because these RNAs have that dual ability to both be a genetic carrier of information as well as have catalytic function similar to proteins. Now, the other thing to keep in mind here is that, as we've mentioned, RNA is less stable. So organisms or entities that use RNA as a genome tend to have a less stable genome. All right. So to summarize this all up, RNA is made of ribonucleotides containing a phosphate group, ribose sugar, and nitrogenous bases. RNA differs from DNA in its sugar-based composition, strand structure, and stability. The three major RNAs involved in protein synthesis are mRNA, rRNA, and tRNA. And finally, some organisms like viruses can use RNA itself as a hereditary material. So with that, thank you so much for attending this lecture, and see you soon.